Welcome. Today, we're diving into GMP expectations for cleaning-in-place systems. I'm Dr. S.K. Singh, and together we'll explore the standards and best practices that ensure these systems meet quality and safety requirements. Let's get started and see what makes cleaning in place both effective and compliant. Let's start with an introduction to cleaning in place, or CIP. It's a vital process in manufacturing, one that cleans equipment without taking it apart or moving any parts. The goal here is clear: remove product residues, wash away cleaning agents, cut down on microbes, and stop cross-contamination in its tracks. All of this keeps the equipment ready to go. From a GMP perspective, CIP ensures that equipment stays in a validated, clean state, always fit for its intended use. When it comes to regulatory guidance, there are several key organizations we need to keep in mind. These include the FDA in the United States, the World Health Organization, and the European Commission. We also look to groups like the Pharmaceutical Inspection Cooperation Scheme, the International Council for Harmonization, and the International Society for Pharmaceutical Engineering. Across the board, the expectation is clear: manufacturers must prove their cleaning-in-place systems reliably meet strict acceptance criteria every single time. Let's start by understanding what a CIP system really is. CIP stands for cleaning in place. It's an automated way to clean equipment without taking it apart. Instead, cleaning solutions flow through the equipment and pipelines, doing the job from the inside. This method is common for things like mixing vessels, reactors, bioreactors, storage tanks, transfer lines, and even water for injection loops. The benefits are clear: less manual work, more consistent cleaning, lower contamination risk, and faster turnaround times. It's a smart, efficient approach to hygiene in processing. Let's talk about what good design looks like for CIP systems in a GMP setting. First, the surfaces that come into contact with the product need to be stainless steel, specifically SS316L, with a very smooth finish, usually less than 0.8 micrometers roughness. We want to avoid dead legs, make sure everything drains completely, and that spray coverage is thorough. Sanitary fittings and valves are a must. Regulators focus on how well equipment can be cleaned and how it prevents contamination. When design falls short, cleaning failures are almost guaranteed. Let's walk through a typical clean-in-place cycle. It usually starts with a pre-rinse to remove loose soil, followed by an alkaline wash to break down fats and proteins. Then comes an intermediate rinse, and if needed, an acid wash to tackle mineral deposits. After that, a final rinse clears away residues, and sometimes sanitization is added before drying or releasing the equipment. Throughout this process, we have to carefully control key parameters: temperature, flow rate, contact time, chemical concentration, and turbulence. Each one needs to be validated to ensure the cycle works effectively. When it comes to cleaning, there are a few critical process parameters we have to keep a close eye on. These CPPs need to be monitored, documented, and maintained within validated operating ranges, no exceptions. Temperature helps break down residues. Flow velocity stirs things up, creating turbulence. Contact time makes sure the cleaning actually works. Detergent concentration boosts the chemistry. Spray coverage guarantees every surface gets wet, and rinse volume flushes away leftover chemicals. Together, these key parameters keep the cleaning process reliable and effective. When we talk about cleaning validation, we're focusing on meeting strict regulatory requirements. That means having documented proof that the cleaning process consistently meets specific acceptance criteria. To do this, we carefully select the worst-case product and equipment scenarios, design a solid sampling strategy, and conduct recovery studies. We also validate the analytical methods and justify our acceptance limits. The ultimate goal here is to show that the cleaning-in-place process is both reproducible and robust, every single time. Let's talk about acceptance criteria and the HBEL approach, a modern regulatory expectation for setting residue limits. We use health-based exposure limits to make sure any leftover residues are safe. Key concepts here include PDE, which stands for permitted daily exposure, ADE or acceptable daily exposure, and MACO, the maximum allowable carryover. For example, if a previous product has a PDE of 10 micrograms per day, and the next product's daily dose and batch size are known, we calculate cleaning limits to keep patient exposure below those safe thresholds. Let's look at the methods we use to check cleanliness. Swab sampling works best for those tricky spots, hard-to-clean areas, places where sprays don't reach, and gasket interfaces. Then there's rinse sampling, which is ideal for large vessels, pipelines, and surfaces that are tough to access. Visual inspection is a must under GMP standards, but the best approach, combining swab and rinse sampling. Together, they give us the highest confidence that cleaning was truly effective. When it comes to data integrity, especially in cleaning records, we have to follow the ALCOA plus principles. That means every piece of data must be attributable, legible, contemporaneous, original, accurate, complete, consistent, enduring, and available. These aren't just good practices. They're required by regulators like the FDA, MHRA, and WHO. If cleaning records fall short, it can put entire validation studies at risk. So, getting this right isn't optional, it's critical. When quality systems fall short, the consequences can be serious. We're talking about FDA Form 483 observations, warning letters, and findings of GMP noncompliance. These issues can even lead to product recalls, something every company wants to avoid. Common deficiencies often show up during inspections. Things like inadequate spray coverage studies, poor management of dead legs, or missing worst-case justifications. We also see problems with rinse sampling, recovery studies, and unvalidated cleaning parameters. Insufficient trend analysis and sloppy documentation practices round out the list. These are the areas we need to focus on. We've successfully validated the CIP process for routine use in syrup manufacturing. Here, what we found. Product residue was well below the limit, at 1.8 micrograms per square centimeter, compared to the maximum of 5. Detergent residue stayed low, just 3.2 parts per million, under the allowed 10. Visual inspections confirmed cleanliness, and microbial counts were safely within limits. This validation was done on a 5,000-liter syrup preparation vessel, using an alkaline detergent wash followed by hot purified water and a final rinse. The results confirm the process is reliable and effective. Let's look ahead at where CIP systems are going. Smart cleaning technologies are transforming how we maintain hygiene. Automated CIP skids handle cleaning with minimal human input. Conductivity-based rinse endpoints and TOC monitoring ensure every rinse is thorough. Real-time cleaning verification and digital records give us instant confidence and traceability. AI helps optimize cleaning cycles, while predictive maintenance keeps equipment running smoothly. All this ties into Industry 4, connecting systems like never before. The future, continuous cleaning verification, moving beyond just periodic checks to constant assurance. Let's wrap up by looking at the key expectations for cleaning-in-place systems under GMP. We need hygienic equipment design and validated cleaning cycles that work every time. Acceptance criteria should be risk-based, with residue limits grounded in science. Sampling strategies must be effective, documentation thorough, and data integrity maintained. Continuous monitoring and review keep everything on track. Ultimately, CIP systems must reliably remove residues and contaminants to meet strict regulatory standards, ensuring quality and safety throughout the product's life cycle. Thanks for joining me today. If you want to explore this topic further or have any questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.